The other important part of a eukaryotic cell is the endomembrane system. And this is what allows eukaryotic cells to be 10 to 100 times bigger than the prokaryotes. Okay, so this system is all about transporting stuff that's produced around the cell. Okay, we've already looked at some of these in our plasma membrane, so we've already seen vesicles. Um, so we're going to see them again. We're going to talk a little bit more about where they come from and what their jobs are. Okay. But there are many components to the endomembrane system. So we've got the one that everybody forgets about, which is the plasma membrane. Because we think endo, oh, that means inside, that we forget that the exterior, that the actual plasma membrane that defines the cell's size and shape and is semi-permeable and all those great things that we talked about in the last chapter, that this is actually part of the endomembrane system. This is where all of these organelles were derived, were from that infolding um, to give us the nucleus and then this endomembrane system. Okay, so again, plasma membrane, really important. The next thing on the list, the nuclear envelope, again, double phospholipid bilayer. We talked about that just a minute ago. Okay, then we also have the endoplasmic reticulum. There's two flavors of that. There's the rough one and the smooth one. We'll get into the details of that. The next thing that we have is the Golgi apparatus. This is kind of, you can think of it as the Amazon packaging center. So all the raw materials are coming in, it's sorting, labeling, and then sending out the vesicles and vacuoles, which are like the little delivery trucks that are taking the crap that you brought from Amazon to your actual house. And then we'll talk about lysosomes that we find in animals, and then how plants do the same function because they already have something else that they use. All right, so let's get into the details of this. So the endoplasmic reticulum, or the ER, is this complex, uh, highly flattened, really large surface area to volume ratio. It's these channels and interconnections that's going to allow this interfacing between the ER and the cytoplasm. Again, I mentioned that there's two different kinds. There's the rough endoplasmic reticulum, um, and the smooth. The rough looks rough because it has ribosomes attached to it, to the exterior of that membrane, so the side that's facing the cytoplasm. Okay, when we look at this under a micrograph, all these little black dots that we're seeing along here, those are all the ribosomes. So you know what ribosomes do. You remember already that ribosomes produce proteins. So the job of the RER is going to be linked to its structure, right? So the form follows function. So the reason that it has ribosomes attached to it is because its job is to quality control, package, and modify proteins, right? So those ribosomes are producing the proteins and they're transferring from the cytoplasm into the ER, right? So from that cytoplasm, boop, into the inner parts of the ER. The ER is modifying, packaging, determining, like doing quality control. Is this a protein that we meant to make? Yes. Okay, cool. Send it off. No. Okay, we need to go break it down. Okay, that's what the rough ER is doing. The smooth ER is continuous. Again, we're tracing from the nuclear envelope to the rough ER, and now we're going to go into the smooth ER. The reason that it looks smooth under this micrograph is because there's not ribosomes attached to it. So if there's not ribosomes attached to it, then what we're going to see is this is where we're going to be modifying the glucose that's made in the mitochondria. We're going to look at that in a minute. And we're going to be modifying those carbohydrates. We're going to be producing lipids, modifying lipids, and we're storing calcium ions. And calcium ions are really important for signaling. Okay. So again, rough ER, proteins, smooth ER, carbs, and lipids, and storing calcium ions. Okay, all that stuff that gets quality controlled and produced from the rough ER and the smooth ER gets sent to the distribution center, which is the Golgi apparatus. Now, almost every textbook that I've seen says that the Golgi apparatus looks like a stack of pancakes, which I guess it does if you're looking at a stack of pancakes from the side. I don't know. I don't really see it, but whatever. All right. I think of the Golgi again as the Amazon packaging center. So the raw materials, right, from the manufacturers, the rough and smoothie are in this case, are sent to the distribution center. The distribution center sorts and packages all of those raw materials and then sends them off to be delivered to the end user. Okay. Um, this is also where organisms that have cell walls, this is where those cell wall components are going to be put together, okay, and manufactured as in the Golgi. Okay. The Golgi are going to be constantly pinching off parts of them to form these little vesicles. So these little transport things that we saw in phagocytosis and pinocytosis and receptor-mediated endocytosis and exocytosis. So remember, like we're just connecting these two chapters, right? And that's going to transport those raw materials. So let's have a look at, so those are our vesicles and vacuoles, okay? Those are the delivery trucks. What we see in animal cells are specialized vesicles called lysosomes, okay? And they are full of digestive enzymes that are going to, so in this case, here we're looking at phagocytosis, okay? So this organic material is being brought into the cell. It's the plasma membrane pinches off a portion of itself to form this vesicle. That vesicle fuses with lysosomes. And then we see this kind of like, right, so this bigger vesicle where the actual digestion of those enzymes are breaking down that organic material. Okay, we can also see lysosomes being used to recycle the building materials for damaged or worn out organelles. So in this case here, we have a mitochondria that's done its job. It's going to fuse with a lysosome. The digestive enzymes in the lysosome are going to break it down. So then we can have those raw materials returned. So that's kind of the recycling center. Typically inside of lysosomes, we tend to see a low pH, which is really good for pulling apart all those monomers, or all the polymers back into their monomers. Okay. The reason that plant cells don't have lysosomes is because they use the large central vacuole for the same job. So things that need to get digested go to the large central vacuole. The vesicles are going to fuse with that. Those old worn out organelles are going to fuse with the large central vacuole. It's a plasma membrane bound organelle. In plants, it can take up to 90% of the volume, so it's really quite a large structure. It's usually full of water or proteins, waste materials, right? So it's just kind of the catch-all of the large central vacuole. This is also the organelle that pushes up against the rigid cell walls to give it to give cells that turgor pressure, right? So that we can stack them together without plants being droopy when we forget to water them. Okay. Peroxisomes are another kind of special vesicle that are found in plants, animals, fungi. And what they're going to do is that they're going to find and isolate hydrogen peroxide, so that's the H2O2, and break it down into water and oxygen. Okay. In animals, we see a lot of these in the livers and the kidneys, where we're processing our waste. Okay. That's all for now.